My name is Nurul Ain binti Muhammad Niki. My number matric 23 DBT 20F1012. I'm from DBT 3A. Today, I will present the lab simulation DNA extraction. First of all, I will explain about DNA extraction. DNA extraction is a routine procedure used to isolate DNA from the nucleus of cell. When ice-cooled alcohol is added to a solution of DNA, the DNA precipitates out of solution. If there is enough DNA in the solution, I will see a streaky white mass. Several different types of DNA extraction methods are phenylchloroform isomer, alcohol, proteinase K, CTAB method, spin column-based method, and magnetic bead-based technique. However, which method to use depends on the sample types and purity and yield of DNA obtained. In this experiment, I need to isolate some DNA from a human test subject. This is genetic testing. For the identification, analysis of forensic evidence. Scientists isolate DNA for a variety of reasons, some of which include genetic testing, body identification, analysis of forensic evidence. DNA extraction is typically the first step in a larger laboratory process. DNA extraction is an important part of the process because DNA first needs to be purified away from protein and other cellular contaminants. We need cell because that's where the DNA is inside. Almost every cell in our bodies is a nucleus, and inside each nucleus is about 2 meters of DNA. First, we need to collect some cells from our test tube. The skin on the inside of our mouth loses thousands of cells every day. These are the cells we are after. From this experiment, we have 4 steps, which is first, collect check cell. Second, burst cell open to release DNA. Third, spread DNA from proteins and debris. And lastly, isolate concentrated DNA. Here are the method and equipment for DNA extraction. This is warm water bath. This microcentrifuge. This is micropipette. This buccal swab. This is sample tube. This lysis solutions. This concentrated salt solution, this resuspension buffers, this is ethanol, and lastly, isopropyl alcohol. First, we have dragged the swab over to the inside of the test tube mouth to collect a sample. Look closely at the swab. Note that it's covered with hundreds of tiny check cells. Inside each check cell is a nucleus, and inside the nucleus is DNA. Using the micropipette, add some lysis solution to the tube. Lysis is a Greek word that means to spread. Place the tube into the warm water bath. The lysis solution contains two important ingredients, detergent and enzymes clad proteins K. The detergent disrupts the cell membranes and nuclear envelopes, causing the cell to burst open and release their DNA. The DNA is still wrapped very tightly around protein cell. Hydness and proteins K cut apart the histones to free the DNA. Next, the cell have stayed in the warm water bath long enough for the DNA to be freed from the cell, and we have removed the swab from the tube and some concentrate salt solution to your tube. The salt causes protein and other cellular debris to clump together. Next, place the tube into the centrifuge. In order to balance the centrifuge, a tube containing water is placed opposite your tube. Click the light to close the centrifuge and turn it on. Inside the centrifuge, the tube spins around at high speed. The heavy clumps of protein and cellular debris to sink to the bottom of the tube, while the strands of DNA remains distributed through the liquid. After that, use the micropipette to carefully remove the top liquid which contains DNA, and place it into a clean tube. The proteins and other cellular debris stay behind. add some isopropyl alcohol to the tube. Inverting the tube several times, mix the isopropyl alcohol into your DNA solution. Because DNA is not soluble, does not stay dissolved in isopropyl alcohol, it comes out of solution. You can now see the clump's DNA with your naked eyes. Place the tube into the centrifuge and click on the light to close it and turn it on. This time, after the sample spin in the centrifuge, the DNA sinks to the bottom of the tube. Once the liquid is removed and the DNA is allowed to dry, can redissolve it in the solution of your choice. Can store it in the freezer for many years, or can move on to your next experiment. That's all for DNA extraction simulation experiment. Thank you.